Okay, so let's see. I'll go over the syllabus, get through chapter 3, maybe start a little bit of chapter 4 today. That's the plan. So here's the syllabus. Today is March 1st, and there was no quiz due this morning. There will be one due Wednesday. That is, looks like the third reading from Master and his emissary. I will be posting, not today, but I'll try and get to it tomorrow, another video about the second reading, the second section of the Master and his Emissary that you've read right here. I've mentioned before. Some of you I know have watched the first one that I already made. The goal of these is I'll talk a little bit about the chapter, and each time I'll try and give you some hints, guidance, some different strategies for thinking about paper topics and how to write the paper. This is kind of like a short lecture about each chapter, and kind of helping you think about the paper with respect to that chapter. And so I will be posting one of those for the second reading assignment that you had this week. I'll try and start on a Tuesday, probably somewhere between Tuesday and Thursday. I'll have it finished and posted. Otherwise, yeah, you do have that quiz due Wednesday, and then I will be posting today the textbook chapter 5 quiz on the sensation chapter. And I will mention chapter 5 is when start chapter 5 and later the difficulty and the length of the chapters increases, at least in my opinion. And so I do want to remind you that for the quizzes, the approach is it's probably better to not just read the entire chapter. I know teachers don't usually say that. The chapters are long, and I don't want you to necessarily read the entire chapter. I want you to look at the questions and the quizzes and read those sections that pertain to the questions. Read those parts of the chapter that pertain to the questions that are asked. Because the questions that I ask in quizzes are the ones that are most likely to show up on in-class discussion questions later on, and of course tests later on as well. So one of the goals is the Blackboard quizzes, as with respect to the textbook, is to kind of draw your attention to those concepts which are most important, the ones that we are likely to cover in lecture, and then in different in-class discussion assignments, and then finally on tests. So we do have one of those. Chapter 5 quiz will be due a week from today. Any questions or comments about assignments? Okay, so as I mentioned, the goal of today will be to finish talking about chapter 3. We may get a start on chapter 4. This chapter 3, is talking about methods of neuroscience, and most of the methods that we've already talked about and that we'll talk about are methods that can be used with humans. Shown here, I've told you we'll be focusing on the functional methods, because these are the methods that allow you to measure brain activity in different mental states or different behavioral tasks. And these functional methods, you're always comparing brain activity between at least two conditions, and those two conditions represent two different mental states, thinking about two different things, or at least two different behaviors that you are performing, compared with the structural methods, which are just like anatomical pictures of the brain. And the structural methods, you're not performing a task, it's just looking at the brain anatomically. So these are the methods which you can use to study the activity of brain activity in humans, because they are not invasive. The machines or the tools that are used to measure the brain activity are always outside of the head. They're always outside of the person's body, outside of the head, outside of the scalp. With the EEG, I guess the electrodes are actually on top of the scalp, but otherwise for the other methods, usually the tools, the things that are measuring the brain activity are not even physically touching the person's head at all, with the exception, of course, is EEG, where the electrodes are actually attached to the person's scalp. So these are the techniques which can study brain activity in humans. We'll finish talking today about EEG, and we'll talk about MEG. We've already talked about fMRI and PET a little bit. So we'll finish talking about these 4 functional methods. Then we will talk about some newer methods, some that could only be used in animals. So these would be the methods where they're more invasive. We'll talk about those later on in the chapter, where you might actually stick an electrode into the animal's brain. So those are invasive, and you can get very precise readings that way. But obviously, that's not something you can usually do with humans unless they happen to be having a brain surgery. So we'll finish talking about these. Then we'll talk about one or two invasive methods and a couple newer methods, which are powerful, but they're still kind of in there. That people are, they're not widely used yet. So that's the plan for today. I do, I guess, want to review just a couple things. fMRI, I'll remind you that this gives you these nice pictures and these the brain activity right here. This is, I'll call it generically brain activity, but maybe you guys can go ahead in the chat. Tell me, what do these, more specifically, these areas of activation, can someone tell me what are in the chat? What do they represent? I'll give you a second to think about this. What are these little colored sections right here? What do they represent? I got some answers here, so we'll take a look. Yeah, oxygen, bold, measures of hemoglobin activity. Yeah, these are all in the right ballpark. Um, the bold response basically is measuring the oxygenated hemoglobin in different parts of the brain. So I'm kind of, some person that just said hemoglobin, that's a little bit vague. Another person just said oxygen. That's probably a slightly better answer. If you kind of combine them, the oxygenated hemoglobin is more specifically what these different colored sections are showing. And the idea is those regions of the brain are which are most active have the most highest metabolism, and then they'll have the most amount of oxygen going to those parts of the brain, most blood flow, oxygenated blood, oxygenated hemoglobin. So in this case, for fMRI, oxygenated hemoglobin is basically a proxy, or it represents activity. That's what it represents. Activity is basically blood flow, oxygenated blood flow, in particular, oxygenated hemoglobin. Good, yeah, the difference in bold between conditions. That's a really important thing to note. Technically, all of these colored things right here are differences in bold response between at least two conditions. You're taking the amount of bold response in one condition minus the amount of bold response in another condition. And then those regions of the brain that have the biggest difference in the amount of oxygenated hemoglobin, the amount of bold response, are the ones where these colors appear. Otherwise, all these other areas of the brain where there are no colors, those are parts of the brain where there was no difference in bold response when people were performing a task. Only these colored differences, only these colored areas are the parts of the brain that were differences when people were performing the task. So good job there. It looks like we're getting that. PET, positron emission tomography, pretty similar as far as I don't have a picture here, but it gives you very similar pictures as fMRI, and it measures kind of a similar thing, which is the amount of blood flow, in particular, those regions of the brain that have the most blood flow. The radioactivity will build up there, and those regions of the brain which release the most radioactive activity are thought to be the parts of the brain where there's the most oxygenated blood going to them. So PET is different. The main technology here is using a radioactive tracer, which attaches to oxygen. And then the radioactive tracer builds up in different parts of the brain. But once again, the whole idea is you're still measuring metabolic activity. Those parts of the brain that have the most oxygenated blood going to them are the regions of the brain that will end up releasing the most radiation. And then the radiation is what is measured by the machine here. fMRI uses magnets, magnetic fields, and those pulses to measure metabolic activity. PET measures metabolic activity with a radioactive tracer. Moving on. EEG, this is the one I think we kind of ended up talking about last time. It measures electrical activity. Once again, you can see that the electrodes on this person are physically touching the scalp. And last time we were talking about frequency analysis. I think we have some pictures. Yeah, let's see. So we'll start with this one. This is like the raw EEG. So this is just like the electrical activity that's being measured at one electrode. This represents one channel or one electrode right here. These are kind of the electrical fluctuations that occur. On the x-axis, this is just time. So this is just the fluctuations that are occurring over time at a given electrode. And for instance, last time I gave you the example, and it would be, it was on the last recording. So I do, if you haven't watched last time's lecture, I do suggest you take a look. I'm trying to think of, yeah, actually it's right here. So the link right here, I'm not going to show it again today, but this is a pretty helpful video to watch as far as frequency analysis is concerned. We have the raw EEG right here. And then these markers represent conditions. And so this is some condition right here. You're measuring the brain's response to a condition here. And then this marker right here, this represents another condition. And if we're performing frequency analysis, what you're doing is you're taking this raw EEG, just the electrical activity as measured on the scalp at one electrode, and then you're breaking it down into one or more different sinusoidal components. What's shown here, this is taken from BioPack, which is the program we use in 625 here, is this is showing alpha activity for this exact channel. So you're using mathematical formulas and you're transforming basically this raw electrical signal right here, and you're kind of filtering it so that you're only looking at sinusoidal activity which is occurring in the alpha range. Let's actually look. Alpha range, once again, is right here, 8 and 12 hertz. This is like a medium frequency. And so remember, when we're talking about frequency, like we're talking about the amount of peaks and troughs that occur per unit time. For alpha band, there's about 8 to 12 up and down motions in a wave per second. That's what 8 to 12 hertz means. It means 8 to 12 waves, 8 to 12 peaks and troughs per second. So you're taking all this tons of information here, and you're just looking, when you're looking at the alpha band, you're breaking it down into those sinusoidal components which are oscillating or have waves of 8 to 12 hertz. And then basically the bigger the height here, that is the higher the amplitude of the alpha band at that point in time. So right here, tons of alpha activity, lots of alpha activity. And then for whatever reasons, and you can see, especially like right here, whatever this condition is, you can see that the amount of alpha activity is really small right here. It's really big for this trial. It's really small for this trial. So I don't actually remember what this was taken from, but you could imagine using this example right here. We had eyes open and eyes closed. We mentioned last time that when eyes are closed, alpha activity, alpha amplitude tends to be really big. So you can imagine right here where the alpha amplitude is really big. This could be when a person's, this whole thing right here from this marker to the end, this could be like when the person's eyes are closed because we have really big alpha amplitude. And then right here, this eyes closed. And then right here would be maybe when the person's eyes open, because you can see the alpha amplitude gets smaller right here. One of the things I really want to emphasize right here is this alpha channel right here. This is all coming from the raw EEG. We're taking this raw EEG right here, which just kind of just looks random, noisy. But then we're using mathematical formulas or filters, and then we're kind of taking out, abstracting the alpha activity, the sinusoidal activity here between 8 and 12 hertz. And then the bigger the height here, the bigger the dispersion, the bigger the alpha amplitude. So here in this one example could be really big alpha amplitude because the person's eyes are closed here. For this next condition, this next marker, relatively smaller alpha amplitude because the eyes are open. So that's kind of some review. I think where we left off last time, we were starting to talk about sleep a little bit. So yeah, I guess this is still a tiny bit of a review. Like when we're awake, the EEG, that is the frequency activity, tends to emphasize, or to the amplitude of the high frequency bands tends to be, high frequency bands tend to be most prominent when you are awake. So right here, the person's awake. And so this picture shows a lot of beta activity. Well, it's not shown here, but which is also true. There's also be a lot of gamma activity. Let's look at these real quick. Gamma and beta, these are the two highest frequency bands, and they're really fast movements. And these are the frequency bands which are most prominent in the EEG when a person is awake and, engaged in particular. But then when we start getting calm, like for instance, when your eyes close, the medium and lower frequency bands become more prominent and the high frequency activity kind of gets quieter. The amplitudes of the high frequency activity goes down, where the amplitudes of the medium and low frequency bands increases, particularly when you just close your eyes, you start to get calm, alpha activity becomes more prominent. That's what I just showed you on that previous slide. You kind of close your eyes, alpha activity gets big. It's kind of like you get calm and then alpha starts to increase. The gamma and beta starts to decrease, but the alpha, which is a medium frequency band, starts to become more prominent. The amplitude of the alpha increases. But then when you actually fall asleep, theta, which is now getting low. Let me show you theta real quick. Theta is right here. This is a low frequency band, and then below that is delta. So theta is 4 to 7 hertz, so we're getting lower, more and more slow as we go from theta to delta. So stage 1, theta becomes more prominent. And then as we get deeper and deeper into sleep and finally get to stages 3 and 4, that's where delta waves are very prominent. Delta waves are basically the lowest frequency wave between like 1 and 4 hertz or so. So relatively low frequency oscillations, low frequency waves that are most prominent in this deep stage of sleep. And let's see if you guys can remember this. Behaviorally, how did I define last time how the deep sleep? What is behaviorally the main difference between deep sleep and not deep sleep? Can you guys put that in the chat? What is the main difference between deep sleep and not deep sleep behaviorally? It's kind of the operational definition. Let's see if anyone's got one answer here. Yes, this is good. Yes. So we got one response and this is correct. How hard it is to wake a person up. Yeah, there we go. Now we got the second couple people getting it here. That's the main difference. In stages 3 and 4, deep sleep, it's difficult to wake a person up. You would need to yell louder or shake them harder in stages 3 and 4 to wake them up compared to stage 1, when it's relatively easy to wake them up. So in general, the harder it is to wake a person up, the more low frequency activity like delta you're likely to find in their EEG. The exception, though, would be REM sleep, which is, as far as waking a person up, the most difficult stage of sleep to wake a person up. And it's also the stage of sleep where people dream the most. But what's paradoxical or surprising about REM sleep, it's a very deep stage of sleep in the sense that it's hard to wake people up. But if you look at the frequency activity, the EEG, the sinusoidal components, you will see that the frequencies which are most prominent are the high frequencies, such as beta in particular, maybe even a tiny bit of gamma, but beta is particularly prominent during REM sleep. So this high frequency component, it's really deep stage of sleep, but this high frequency component is prominent. Presumably that's some type of neural correlate of dreaming. And dreaming is a sense of being kind of conscious even when you're asleep. And so it's interesting to think about this high frequency activity is occurring because of the dreams, which are something you're kind of conscious of while you are asleep. So REM sleep is paradoxical because it's a very deep stage of sleep, yet high frequencies like beta are most prominent in the EEG. And I really never talked about this one, and I don't think I'm going to spend time. So this is really the thing which is most important for you guys to remember, to understand. Okay, so now we're definitely getting into new stuff. And I want to point out, I have added 1 or 2 slides for this chapter since last time. This one was in the original slides I posted last week, but this slide right here, which I'm going to talk to you about in a second, I did add this, and I uploaded new slides with that, a new slide file with that new slide coming up. So now talking about event-related potentials, and we've talked a lot about EEG already. And when we talked about EEG so far, we've been talking mostly about frequency analysis. You take in that raw electrical signal measured at one electrode, and you're breaking it down into its sinusoidal components. And we're comparing the amplitude usually of those of 1 or 2 sinusoidal components between different conditions. Kind of the classic example I've gone to several times now is alpha amplitude tends to be bigger when your eyes are closed, when you're calm and relaxed. And alpha amplitude is relatively smaller when your eyes are open, when you are awake and conscious. And so for frequency analysis, we're comparing the amplitude of different frequency bands between at least two different conditions. We're always, for these functional methods, doing the same thing. We're measuring brain activity, and we're between two different conditions. That's what we're always doing for fMRI, PET, EEG. We're measuring brain activity and comparing it between at least two conditions. For frequency analysis, the brain activity we're using specifically is the amplitude of some frequency, like alpha, and we're comparing it between the two conditions, eyes closed, eyes open. Now, event-related potentials they're another way of using EEG. So when I say EEG, all that really is that is the method for recording electrical activity from the scalp. Frequency analysis is a specific way of using EEG, where your frequency analysis, once again, you're breaking down the raw EEG, the electrical activity, into those different sinusoidal components. And that's one way of using EEG. Another way, event-related potentials, you are not breaking down the EEG into the different sinusoidal components. Instead, with event-related potentials, I guess this definition here is kind of help you. They're precisely time-locked analysis of the raw EEG signal relative to the onset of some event. And so I say onset, I mean like the appearance of something. That's usually the onset, or it could be you hear a sound. That is the onset. When something occurs, something appears, or you start to, or something like you hear something, or something appears visually. That is the onset. And we're measuring that EEG, the raw EEG response, to the onset of at least two different things. And the thing which is important about ERP, or to keep in mind, is precisely time locked. We're looking at differences in the EEG, the electrical activity, usually at a pretty specific point in time. So I'll illustrate this with the N170 event-related potential, which is shown right here. The two things that are being compared, we're comparing brain activity between these two things right here. Basically, you're either looking at a face or you're looking at a car. Those are the two different mental states. My brain, when I'm thinking about a face or when I'm looking about a face, my brain when I'm thinking or looking at a car. How is the brain different between these two different mental states, two different behavioral states? And so this is like the N170 ERP right here. And it's kind of important to decompose this into the different, parts. So right here, time point 0, you can see that says 0 right here, the intersection of the y and x-axis right here, time point at 0. This is the onset of one of these two stimuli, one of these two pictures. You're at this point, time point 0, you're either a face appears on a computer screen or a car appears on the computer screen. And then as we go to the right, this is time. And a very short amount of time at that. This whole thing is only 400 milliseconds. So this whole thing is less than 1/2 of a second. This whole thing is, 2/5 of a second. And then we see these two different waves right here, these two different graphs. The dark colored one right here, this is the brain's response to faces. The lighter colored one, the light gray, this is the brain's response to cars. So on trial 1, there's a face that occurs, and then the brain responds, and this is what the brain's response looks like. This right here, though, the dark line right here, this basically is if you're taking the brain's response to a face over like 100 different trials, and then you average the brain's response for those 100 trials. That's what this dark one looks like right here. This is basically 100 trials or so of looking at a face. This is the brain's response. If you average the electrical activity that occurs 100 times, look at a face, you record the brain activity. You look at a face again, you record the brain's activity again. You take those 100 times where you're showing the brain's response to a face, you average them together, and this dark line right here is what you get. You do the same thing with cars. You show a car 100 different times. Each time you're measuring the electrical activity at some electrode on the brain. You do it like 100 times, you average those 100 trials together, and eventually you get this waveform, which represents the average of 100 conditions or so. And this is a typical way of studying event-related potentials. Looking here at the differences in average amplitude. So that's what the y-axis is right here, typically measured in microvolts. This one happens to not be labeled, but microvolts is the typical measurement. A measure of electrical activity. Microvolts is the y-axis. At specific time point relative to a stimulus. So that last point, specific time point, this is an important component of the event-related potentials. Once again, precisely time-locked, a specific time point. And then the specific time point here is, you can see that these differences occur. There's pretty notable differences here. And then we have this arrow, it's saying in 170, and it's pointing right here. This right here, this is the in 170 ERP. It is the N right here stands for negativity. And if you look, there's a negative sign right here. The up is positive in this graph. There's a negative, the down is negative. And so that N comes from the fact that if we look at the direction of these waves right here, they're in the negative side. So negative voltage, a negativity. That's what the N stands for. The 170 stands for milliseconds. Time point 0 right here, but then we're going forward in time, forward in time, forward in time. 170 milliseconds after the onset of a stimulus. Once again, that stimulus is either a face or car. There is a negativity for both. There's a car, you can see there's a negativity in response to the car. You look at the face, you can see that there's a negativity in response to the face. The N170 is the finding that at 170 milliseconds after the onset of the stimulus, there tends to be a bigger negativity in response to faces than to any other type of object. In faces in particular, there tends to be a big negativity in response at 170 milliseconds after the onset of face relative to other objects like cars. So like you can kind of think of cars are like the control condition. Faces are generally the experimental condition. The negativity at 170 milliseconds after the onset of the stimulus is bigger in response to faces than it is to cars. And this is important. Last time someone was asking, so what does this stuff all mean? That's still kind of debated exactly. Like it's not really known where in the brain this electrical activity is coming from. But this type of thing is important because there are individuals, for instance, people with prosopagnosia, which is face blindness. And if you look at their event-related potential responses to faces, they're usually abnormal. They look different than this. And so you can use these electrical responses to, you kind of know this is what a typical brain will look like when it's responding to a face. And then you can use these event-related potentials to see if people's brain responses are abnormal compared to like a normal brain response. So I have this is what I added, is a video that actually shows the measurement of some event-related potentials. I remember when I was an undergraduate, I was always confused about the differences between, like, I had these terms in my head, EEG, frequency analysis, event-related potentials, and they're all kind of this like, it was difficult for me as an undergraduate to distinguish them. And so I'm thinking that playing this video right here will help kind of distinguish the difference between event-related potentials and frequency analysis. Both, let me remind you, both frequency analysis and ERPs, frequency analysis and event-related potentials, both use EEG. EEG is just measuring activity on the scalp with electrodes. Frequency analysis takes that electrical activity, breaks it down into different sinusoidal components. Event-related potentials takes that electrical activity and it measures differences in amplitude at specific time points between at least two different conditions. The N400 is another ERP. Once again, the N still stands for negativity, but now the 400 is here, and the 400 comes from the fact that this ERP that I'm about to talk about, the key place where you're comparing the amplitude between the two conditions is 400 milliseconds after the onset of some stimulus. So the N400 amplitude, or the N400 ERP, as I kind of mentioned here, it measures electrical activity in response to a word in a sentence which either satisfies or violates a semantic expectation. So semantic means it refers to like the meaning of the words in the sentence. And so I have an example here. She grew a plant in the pot. That is expected. There's nothing unusual about that. She grew a plant in the fridge. That's a little bit unexpected. I guess you may be able to grow a plant in a fridge, but she grew a plant in a pot. That definitely is like, that makes sense. There's really nothing unusual about this sentence at all. She grew a plant in the fridge. That's a little bit more unusual of a sentence. So this sentence right here kind of violates your semantic expectation, in particular, this word right here, fridge, up to the point she grew a plant in the, that's a typical sentence. But then the next word that you hear, she grew a plant in that final word can either satisfy or violate your expectation, which is built up from those previous words in the sentence. She grew a plant in the pot. That makes sense. Your expectation is satisfied. In fridge, she grew a plant in the fridge. That's a little bit weirder. Your expectation is a little bit violated. It's kind of unusual to grow a plant in a fridge. And so I'm going to show you, I guess I'll mention the key finding here. 400 milliseconds after the onset of the target word, there tends to be a larger negativity in response to the unexpected word. So here, there's a larger negativity in response to the word fridge compared to the word pot. 400 milliseconds after the onset of the word. And so I'm going to show you how the N400 is measured right here, hopefully. Okay, so it's now measuring. So you can see that these are the raw EEG. So it happens to be there's actually two electrodes here. So the red one, this is like channel 1. This is raw electrical activity on one electrode. And then the blue, channel 2, this is raw EEG, the raw electrical activity measured in another electrode. So here, this is a pretty simple setup. We don't have an EEG with like 50 different electrodes. In this one, we're just using 2 electrodes, keeping it simple. We could have done this just as easily with just 1 electrode. The key, though, there's kind of like 2 things you have to track simultaneously. The stimuli, the words people are reading, will appear right here. And then the electrical activity is occurring right here. I think I have a question. Let's take a look. So the question is this measuring the brain's reaction time and how strong the response is? Yeah, you could say that. I mean, that's a little bit of an oversimplification, but you can think it definitely, when you say how strong the response is, that's when I'm talking about the amplitude of the response. The bigger the brain's response, the bigger the amplitude. And so, for instance, with the face one that I just talked about, the brain's response is bigger in response to faces than it is to cars. There's a bigger negativity in response to faces, 170 milliseconds after the onset of a face compared to cars. That's what I showed you previously. A similar type of thing here for the N400. The brain's response is bigger after the brain's response to a word that is unexpected is bigger than the brain's response to a word that is expected, particularly at that 400 millisecond time point. So yeah, the question is this measuring the brain's reaction time and how strong the response is? That's pretty decent way of understanding this. We're looking at a specific time point after the onset of something, and we're looking at the brain's response, and we're looking at differences in amplitude at a specific time point. So that's a decent way of thinking about this. So I'll continue on here. So you have to kind of look at this right here, and then I'll pause it at strategic times. I'll kind of read it aloud. I'll read it aloud. So brain's going. She grew a plant in the pot. So an important thing just happened. Right here, this is an event marker. I showed you a different event marker for earlier when I was showing you the eyes closed, eyes open stuff earlier. we had event markers there. And that earlier one, which is a different setting in BioPack, when the person's eyes closed, there was that little triangle that appeared at the top. And then when the person's eyes opened, there was another triangle that appeared at the top. And those different triangles, those event markers, they were basically saying, this is the part of the brain, this is the brain's response during the eyes closed condition. The other triangle was, this is the brain's response during the eyes open condition. I showed you that last time, and I kind of reviewed it earlier today. For this is kind of a similar situation, except here, instead of using those triangles, this is the event marker right here. And so this thing right here pops up at the precise moment in time that the word pot appeared. She grew a plant in the pot. And as soon as the word pot appears at that exact time point, this is time point 0, basically right here. And so when we're looking at, this is trial 1, this right here is the brain's response to the word that is expected. And so what we would do is we would take this time point, and then 400 milliseconds after the word, we would be measuring the brain's activity right here. So you can't really see, it looks kind of just like noise right here. You can't see a whole lot right here. But what I want you to really focus in on is this is trial 1 for a word that satisfies your expectation. Eventually we'll do like 100 of these. We would add up the brain's response to 100 different trials when of the brain's response to a word that satisfies the expectation. And then we'll also do the same for words that violate. So let me show you a word that violates here. She grew a plant in the fridge. So when the word fridge was presented at that exact time point, this blue marker appears. This right here represents the brain's activity when the word is unexpected. And so the N400, the condition of unexpected words, takes this brain activity and it will add it to the next trial in which there's an unexpected word, the next trial, and you basically take the average brain response relative to this exact time point when the word is unexpected, you average those together and you get a wave. You do the same thing for those trials which the word is expected. So the N400, the negativity 400 milliseconds after the word fridge tends to be bigger than the negativity after the word pot. So we will, I know this stuff can be pretty tough. This is the first time we'll talk about the N400. We will actually review it in a little bit more detail later on in chapter 11, I believe. So that's ERPs. We have 2 different examples, the N170 and the N400. So now we'll talk about MEG. We're not going to talk about this one in nearly as much detail. I spend a lot of time talking about EEG in detail because this EEG is the method that in the psychology department we have the most access to. For instance, a lot of you guys are neuroscience majors, which means you'll take 625 sometime in the future, which is like the capstone course. And you learn to use a lot of physiological methods in that class. And the main brain method that you use in that class is EEG. So I cover EEG more in this course because it will help prepare you for what you'll learn about in EEG in that future course 625. MEG is kind of similar to EEG in some ways, in the sense that both EEG and MEG have really good temporal precision. We can see what's going on in the brain with MEG, just like with EEG, with millisecond precision. We can just tell differences in amplitude that occur between 2 different conditions with really precise, really good temporal precision. There are some differences, of course, whereas EEG measures electrical activity, MEG measures magnetic activity. So there's some magnetic field which is kind of surrounding your skull, and it's presumably caused by things inside the brain. And then in particular, the magnetic field is perpendicular to electrical currents in the brain, and that is important for the ability of the machine to pick up certain types of responses, certain types of magnetic responses. So it turns out not all magnetic activity in the brain can actually be measured by MEG. So it is a weakness of MEG. But nevertheless, it can get a lot of important activity with high temporal precision. And one good thing about MEG is that the magnetic fields which are created and which are measured by this unit, they are not distorted by the skull. And so for EEG, I mentioned that EEG has really good temporal precision, but with EEG, we don't know where in the brain the electrical activity is coming from. With MEG, on the other hand, it has really good temporal precision. And because the magnetic field is not distorted by the skull, with MEG, you can actually localize with some accuracy, with pretty decent accuracy, where in the brain the activity is coming from. So MEG, although it has some limitations, and the main limitation, a couple of limitations. One, it's really expensive. These things are expensive to buy and to operate. They're expensive. Another limitation is that because the magnetic field is perpendicular to electrical currents in the brain, there's only not all of the brain's responses can be measured with MEG. So that's another limitation. But for those brain's responses that can be measured with MEG, they can be very powerful because it can measure the amplitude fluctuations that occur with very good temporal precision, just like EEG. But you can also localize them somewhat to specific parts of the brain. And to kind of illustrate, I've seen some MEG studies, and they're particularly good for like localizing, like along the temporal lobe. You can kind of localize different sections, like you have some type of fluctuation which is occurring along the temporal lobe, which is here on the side. And MEG is pretty good at saying, like, did it come from this part of the temporal lobe, or this part of the temporal lobe, or this part of the temporal lobe? It's for those parts of the brain which are kind of along the surface, like the outside of the temporal lobe. MEG is pretty accurate at localizing exactly where the signal is coming from. It looks like we have some questions. Let's take a look here. So question was, well, I explain what temporal means again. Temporal refers to the idea is that when we're measuring the brain's activity, like we're presenting like an object, and then we're measuring the brain's response to that object. With fMRI, for instance, when you're seeing those colors on the brain, those colors on the brain represents the activity which is kind of averaged over a 10-second time point. That's fMRI. fMRI does not have good temporal precision because we're like just looking at this is the brain's activity over 10 seconds. And it's just all of those things that happen over the 10 seconds are kind of represented by that single blob. But with good temporal precision, we can see the fluctuations in brain activity that are occurring, like with each millisecond that occurs. We can know that at 100 milliseconds, there was a big positivity, and at 150 milliseconds, there was a negativity, and at 250 milliseconds, there was a positivity. So with EEG and MEG, we can see the fluctuations that occur at each millisecond, basically. With fMRI, you don't get to see those fluctuations over each millisecond. With fMRI and PET, you just see like this is what the brain was doing during the entire trial. This is kind of a summation or an average of all the activity over a 10-second window. EEG and MEG, you get to see each fluctuation. That's temporal precision. Good. The other question, temporal is time. Oh yeah, someone was answering that for them. Good. Okay, good. Let's continue on then. Okay, so yeah, so here's when we're getting to an example. We're kind of getting away from human stuff temporarily. So single cell or multi-unit activity or multi-unit recordings, these are basically only done in animals, with the exception of they're kind of can be done in humans if the person is having a surgery in their skull, they're having type of brain surgery and they're going to be awake during the brain surgery. You can actually measure electrical activity by sticking electrodes directly into the brain. And these are like the best, most accurate recordings you can possibly have, because it's getting everything which is going on in that area where the electrode is, and it has really high temporal precision. You get all of the details that are occurring at every single millisecond that you're making a recording. And they also have very good spatial precision. You're sticking an electrode in a specific part of the brain, and all the electrical activity that you're picking up is coming from those brain regions that are like directly adjacent to the electrode. Looks like we have a question here. How does the electrode not damage the brain? They're very small, but they can damage the brain a little bit, but they're small. And so the damage that they do is relatively minor. but they do. I mean, that's one of the reasons. I mean, for humans, you'll usually just put the electrode right on top. You don't usually insert it into the brain. With animals, they'll actually kind of insert it deeply into the brain sometimes. The least invasive ones, though, are when you just place the electrode right on top of the brain. That won't damage at all. But if you do insert the electrode into the brain, it will cause damage. But the small electrodes will have less damage. You're welcome. Um, the main dependent variable for these types of studies, whether it's single cell or multi-unit activity, once again, you can insert an electrode into the brain or on top of the surface of the brain. The main dependent variable is the rate of firing of action potentials. And once again, for all these functional methods, we're comparing brain activity between two different conditions with single and multi-unit activity. You're comparing the amount of action potentials. I mean, we're so close now. We're up like right in the brain. You can literally tell these neurons are firing action potentials right here. They're not firing action potentials. And so that's the dependent variable, the amount of action potentials between at least two different conditions. And by the way, single cell and multi-unit, this just represents the amount of neurons that are presumably being measured. So a single cell, you're trying to isolate just the electrical activity of a single neuron. With multi-unit activity, which is the typical way, you stick an electrode, which is presumably getting the activity of all of the adjacent neurons. It's hard really to isolate a single neuron. Multi-unit activity is a little bit more common than single cell. There is an example right here of a single cell recording. I'm not going to show this video, but it's a pretty classic video, and it does illustrate this idea of single cell recordings. Okay, so brain lesion studies. This is now, I mean, this is another type of study which you can't like randomly assign people to have brain lesions or brain damage, but sometimes, whether in humans, brain lesions occur naturally, because of getting hit in the head very hard, or because of a stroke. Strokes are probably the most common kinds of brain damage that can occur. And as a result of this brain damage, you can look at changes in the behavior that occurred after the brain damage. And then you can kind of get an idea, whatever brain region was damaged, the difference in behavior after the damage compared to the behavior before the damage, that illustrates the function of the brain region. The person was behaving one way before the damage, then they get the brain damage, and then after the damage, they behave a different way. Those differences in behavior between the before and after illustrate the function of the brain region that was damaged. So brain lesion studies are really the most powerful type of functional research you can do regarding the brain. And the main criticism, which if this were like a graduate course, I've talked more about this, but I just will say it because if you go into neuroscience later on, you will see this idea and you will talk about it in more detail. I will just say this is the typical critique or criticism of neuroimaging methods, fMRI, EEG, is that they're correlational in a sense. Compared to with lesion studies, these actually provide like causal evidence. If I lesion the left temporal lobe, and then all of a sudden I can no longer understand speech, that gives me good causal evidence that this region of the brain is important, or it causes the ability, or it is solely responsible for the ability to understand speech. So like the more convincing or causal data, causal findings require that the brain is somehow changed by the researcher, or by a lesion that happens by accident. And so yeah, there's some examples in animals. You actually can assign. This is really common. We'll see some examples, plenty of examples throughout this course. You actually can randomly assign animals to either the control condition where their brains are intact, or a brain damage condition in which their brain is ablated or somehow damaged. Or there's many different ways. They'll sometimes physically remove part of the brain, or they'll inject a drug which will deactivate it or kill the brain cells in a given part of the brain. And then you compare in the animal the differences in behavior between brain intact, brain is not intact. As I mentioned, in humans, really the only way that you can study brain lesions is just when they happen naturally. You can't randomly assign people to experience a brain injury. With the exception of a new technology, which I'll I believe I'm talking about in the next slide, which can actually cause kind of artificial or temporary lesions, temporary brain damage, which will last like 30 seconds or a minute. And then after that, you're fine. So it's kind of an interesting but controversial technology. Yeah, it's okay. So I guess it's in a few slides. So in a few slides in the future, I'll be talking about this artificial lesions. These are the typical way of getting causal data. You have to, as the researcher, you have to cause some type of a change in the brain's activity, either make the brain super working, extra excited, increase the activity of the brain, or decrease the activity of the brain, damage. And then you compare the differences in behavior between those two states. Hold on. Do you always have to use random assignment with animals like you do with humans when using an experimental design? Yeah, I mean, as far yeah, usually I mean, you don't you would randomly pick an animal out of like usually they have like a bunch of rats. When I worked at a previous place, there was like a room with 100 different rats, and you go in and you just pick out a rat, and then what we did is the first rat we would pick out would be the experimental condition. And so that rat would have its brain damaged. And then you would kind of randomly pick another rat, and then that rat would be in the control condition. So we would alternate brain damage, control, brain damage, control. But each time we were kind of randomly picking a rat. So I think that answers your question. I mean, that's what random assignment basically looks like for animals. And I would say, yeah, researchers do try to abide by some type of random selection for even experimental research on animals. So yeah, here's another example of lesion studies. So of lesion findings. So there's this region of the brain. This is the left hemisphere of the brain right here. And this is the temporal lobe right here. This is the occipital lobe. And there's this kind of area right between them. Once again, occipital lobe, temporal lobe. Right here is this region of the brain, sometimes called the VOT, so left ventral occipital temporal. So you don't really know. Ventral just means like in front of or like towards the belly is really what ventral means. Occipital temporal, though, is basically it's on the boundary right here between occipital and temporal right here. This is a region of the brain which its function has been pretty clearly demonstrated by individuals that have had damage to this part of the brain. When people have damage to this part of the brain, it causes a condition known as acquired alexia. And acquired alexia, it's very similar to dyslexia, except dyslexia is thought to be a developmental disorder. You're like, you're born with some with dyslexia, and then basically as you're trying to learn reading, you don't develop typical reading skills because dyslexia is considered a developmental condition. Acquired alexia, on the other hand, means that you had at some point normal reading ability, but then because of brain damage, you now became alexic, which alexia basically is troubles reading, in particular sight reading. So individuals with alexia, like I'm just looking at these words, like various patients, I can just read that without even thinking about it. People with alexia, though, they can't just sight word the word patience. They would have to sound it out, patience, and then it maybe would eventually be able to sound it out as patience. And that's often the way dyslexia is characterized, is by people that need to constantly sound out words. Alexia is the same way, always having to sound out words. You can't sight read them. And that's caused by damage particularly. So this seems to be a region of the brain right here, which is important for automatic sight reading of words. Looks like we have a question. How do they test for, can you be more specific with your question? How do they test for what? How do they test for the ability to read words? For alexia. Yeah, so there's many different tests for it, but one is that they'll just measure the rate at which you read words. They'll just present a word on a screen, and then they'll measure the amount of time it takes you to read the word, like say it out loud. People with alexia are slower to read words than people with dyslexia. People with alexia are slower to read words than controls. People with controls can just read a word as soon as they see it. People with alexia, it takes several seconds. Another thing they do is they actually look at their eye movements, and they find that people with alexia, they kind of focus in on each letter of a word. Whereas people that are controls, they kind of with most people, they actually just look right here on the word, and then they kind of read the entire word like instantly. You just kind of focus here on controls. They'll kind of focus in like towards the beginning, but not the very beginning of the word. they'll focus right here, and they're able to kind of read the whole word just with one eyeball fixation. People with alexia, on the other hand, they have to kind of slowly go left to right. Their eyes are going through each individual letter, whereas controls, most of you, I'd assume, are able to get the entire word with one fixation. You just look at one part of the word, you get the whole thing. Is there a cure for alexia, or is something someone has to live with? There probably is some type of ways you can, like, usually you get compensatory strategies. For instance, well, I know more about dyslexia regarding this question. People with dyslexia have trouble acquiring automaticity when they're reading words. But people with dyslexia will usually find they usually kind of develop instinctually or unconsciously alternative strategies for reading. So for instance, individuals with dyslexia, they actually will usually show less activity in the left hemisphere because the ventral occipital temporal area doesn't work that well. But if you actually look at their right hemispheres, there's more activity in people with alexia and dyslexia. So it's like the other parts of the brain try and compensate. And it's thought that the other regions of the brains that become active after brain damage, or if you have dyslexia and your left hemisphere is not working, these other regions of the brain are attempting to compensate. And because these other regions of the brain are doing different things, it's thought that basically what the person is doing is they're trying to use alternative strategies, different ways of reading. I mean, that's the theory, at least. Different brain regions become active. Those different brain regions are thought to represent like different strategies. And there are therapies that can be used to improve sight reading. Though my understanding is, for the most part, people that if you learn reading well, and then you have damage to this region of the brain, like when you're 20 or 30 years old, your sight reading, your automaticity is probably never going to be as good as it was before the lesion. Does alexia affect you even if you've already learned to read before? Yes, you could. Okay, so an interesting thing is if you're like 5 years old and you get damage to the left VOT. Oh, by the way, I should also mention the VOT is also called the visual word form area. If you get damage to this area when you're only 5 years old, you'll actually be fine, because the right hemisphere of your brain, there's kind of an equivalent area over here. If you're really young when you get the damage, then the right hemisphere of the brain will end up kind of picking up the slack during the development. So if you get brain damage when you're really early, the right visual word form area will actually be able to get really, really strong. And so you'll probably be able to develop pretty typical reading ability if the lesion is really, really early. But if you get the lesion when you're like 20 or 25 years and you already have your reading skills solidified, then your brain is like, all right, this is the part of my brain which I use for sight reading words. The right hemisphere of the brain is now being used for something else. If this region of the brain becomes lesioned when you're 20 or 25 years old, then things are problematic. Like the earlier you are when you have damage to your brain, the more likely that due to brain plasticity, your brain will develop effective compensatory strategies. When you're older and you have brain damage, it's much more difficult for the brain to compensate. Let's see how we're doing here on time. Yeah, I know it's a little bit of a longer lecture, but I really want to get through chapter 3 today. So another example of a lesion study is severing the corpus callosum. We're going to be talking about this a lot in chapter 4. Master and his emissary also talks about this. So I'm not going to say too much other than, well, the corpus callosum is this region of the white. It's a white matter pathway. It's probably better viewed right here, the right hemisphere and the left hemisphere. So here in this picture, this is like looking at the person front on. So this is like, you have to imagine that this brain, their face of this person is looking at you. So this is the person's right hemisphere. This is the person's left hemisphere. In between the two hemispheres, this white matter pathway, you can think of it as like wires that allow communication between the two hemispheres of the brain. And we'll talk more. I'm going to wait about till the chapter 4 until we talk more about exactly what happens when this is severed. But I will say they've learned a lot. They've used lesion studies they've severed this corpus callosum, and then they've looked at changes in behavior that occur as a result of the corpus callosum being severed that have provided information, evidence regarding the function of the corpus callosum. It is quite clear. The corpus callosum allows information to be passed back and forth between the left and right hemisphere. In chapter 4, we'll talk more about the specific evidence that supports this idea. How do you study this in more detail? We will talk about that in chapter 4. And I will say kind of one of the key findings after surgery, most patients can read words when they're presented to the right visual field. But if a word is presented to the left visual field, they will not be able to see it. And that's evidence, and I'll explain it in more detail later, that information is being exchanged between the two hemispheres. TMS, or transcranial magnetic stimulation. This is what I was talking about earlier, artificial or temporary lesions. So obviously with humans, you can't randomly assign people to have their corpus callosum severed, or you can't randomly assign them to have their visual word form area, that is the ventral, occipital, temporal area, removed or damaged. You can, though, cause kind of those regions of the brain kind of temporarily fall asleep. That's how I think of TMS. I kind of think about it like when you are lying on your arm too long and then your hand falls asleep, or if you're like sitting on your feet and then your feet fall asleep. There's like a minute or so where you can't really feel much sensation in that region of your body. There's kind of a similar thing which can go on with the brain, an analogous thing, where you can kind of aim a magnetic pulse into the brain. That's what this little wand thing creates, a magnetic field. And this magnetic field can basically do one of two things. It can either make your brain fall asleep, which is decreasing neural activity, or it actually, if you align the magnetic fields just right, can actually temporarily boost or activate different parts of the brain. But this is really helpful for causal studies because you as the researcher can influence the brain activity. You can either increase it or decrease it. And then we look at, here's brain activity before the TMS activation, or here's behavior before the TMS activation, here's behavior after the activation, and then we can compare the behavior, the before and after, and we can get an idea of what a region of the brain is doing for behavior, and it's causal evidence. So this can be very valuable evidence. I will say TMS, it's difficult to use and it's not published a lot. So it's potentially a very useful method, though there's not a ton of research, and probably because it's hard to use and there aren't a lot of people that are skilled in using it. A similar technology is called direct stimulation. Looks like we got a question here. Is this TMS the same kind used for treatment of depression? Yes, it is. Yeah, it is the exact same thing. Another question. Yeah, okay. So the direct stimulation, this is very similar to TMS. It's basically except usually this is electrical stimulation that is when the brain is open. So the brain is exposed. So it's used primarily in animals, but it has been used in humans. I've actually already talked about an example, the Montreal procedure by Penfield. This was the procedure which Penfield discovered the motor homunculus on the frontal lobe. That's the part of the brain that when it was stimulated electrically can cause motor movements of the hand, or it can cause your head to shake, or it can cause you to shake your foot. Depending on which part of the motor strip was activated, different parts of the body would move. It was the Montreal procedure, is basically direct stimulation. It's electrically activating different parts of the brain and then looking at behavior changes as a result. For the Montreal procedure, they directly stimulated the brain and could cause movements. So that's the Montreal procedure, an example of direct stimulation. Direct stimulation is just electrically activating different parts of the brain by directly touching that part of the brain with some type of stimulatory device. Yeah, that's about all I need to say there. I'm not going to try and push it. We'll call it a day there. I just have a tiny bit more. I'll answer these questions, then I'm going to call it a day. Yeah, actually, those aren't questions. Good. So we'll just call it a day, and I will continue. I will definitely finish chapter 3 next time. We'll get a good way into chapter 4. If anyone wants to send me an email, ask questions and stuff, happily I will answer them. I will see you Wednesday.